Welcome everybody to the second day of the workshop, and I'm very happy to have Patrick open the session with his talk on from big data to systems pharmacology.
Thank you for that. Thanks for the invitation. Very nice. It's actually my first time at this side of the day. So I like it. I'm a bit ashamed because Pascal just told me that my title is boring, but I'll try to make it more exciting. So basically, I will start by saying that these last years, what I'm doing is trying to overcome personal frustration, right? So these are my friends from childhood. And as you can see, all of them are working in companies that have to do with image processing, natural language processing, artificial intelligence, etc. So we're turning 50 this year, which means that we've been around for long enough so that we have witnessed all the progress in this field for many, many years. And in fact, my first paper ever was a neural network in a journal that it was called CABIO, that's Computer Applications in Biosciences, that this was the seat of bioinformatics now, right? And as you can see, it was implemented in Excel. You can imagine that I didn't write a neural network in Excel. I implemented it in C. But then the comment that I got back from the reviewers, like, if you want any biologist to use a neural network, you have to be, or it has to be implemented in Excel, because no biologist will ever use a compiled program. So I did implement it. Anyway, so for the last 30 years or so, I wanted to play with my friends saying, hey guys, I want to use your algorithms, these things so cool that you're doing here. And their answer was always the same. So you do not have enough data. You think you have a lot of data, but you don't have enough data to play with us. Okay, now screw them, because we have more data than they do, right? So now the problem in biology and chemistry is not the data anymore, it's how we format this data. And this is what we're trying to do in the lab for the last years. So let's start with chemistry first. So still, small molecules are the main assets of pharmaceutical companies, as you know. And basically, this is where I come from, right? Bioinformatics, many of you. Proteins, that's very cool. So biology is self-formatted. We don't have to play very much with it. So if you're playing with nucleic acid, it's only 4 letters. If we're playing for proteins, it's 20 amino acids. We have these beautiful domain architectures. We have a sort of clear sequence to structure to function relationship. And all these things have been optimized by millions of years of evolution. And if anything, we only have around 20,000 proteins or 20,000 genes, 100,000 proteins that are organized in beautiful databases. When you go to chemistry, this is a big mess, right? So this really is hell. So these guys are monolithic entities. You cannot split them into functional domains. No clear architecture. They have been, if anything, mildly optimized by one medchem, couple of rounds. And we have more than 200 million commercially available molecules. You can pick up the phone, buy one of these, but the chemical space is huge. I mean, we're talking about 200 billion molecules that could be easily synthesizable here. So, and if anything, you can look at this, you might not know this is RNA Pol 2, but it's pretty clear that there's a template, there's a machine, and there's something that is being copied. You look at this, and you either know exactly what it does, or you cannot guess its function. So what the computational chemists have been doing in the last 30, 40 years is trying to encode somehow the chemical structure of these compounds so that we can compare them, right? So trying to encode this information in the form of a vector, different types, 2D, 3Ds, scaffolds, etc. Because what we know is that whenever two molecules look similar, they tend to bind to the same receptors, have sort of the same targets. Then in the last years, thanks to all these omics techniques that we all love, what we have seen is that not only molecules with similar structures tend to bind the same receptors, but molecules with similar transcriptional profiles tend to have similar bioactivities, or the molecules that kill the same sort of cancer cell lines also tend to have similar bioactivities. So what we set up to do with Miquel Duran in my lab, some 6 years ago already, is trying to encode in the form of a vector, not the chemical structure of these compounds, but their bioactivities, right? And this is exactly what we did in a resource that we call the chemical checker for obvious reasons, that basically is formed by information for 1 million, more than 1 million bioactive molecules for which we have experimental information. So no bioinformatics magics here yet. right? And then what we did is to organize this resource in 5 levels of complexity, following a little bit the rationale of how we understand drug action, right? So in the first level, we have the chemistry descriptors, 2D, 3D fingerprints, scaffolds, etc. Then for a molecule to be bioactive, it has to bind to a biomolecule. This is the second level, the target, mechanism of actions, the genes that metabolize, this is a small molecule, binding, etc. Then when you modulate the activity of one of these targets, this information gets kind of spread out in the cell through networks of different types, signaling networks, interactome networks, etc. And we can capture all this information in cell-based experiments. Gene expression, cancer cell line panel, chemical genetics, high content screens, etc. And finally, for the few molecules that reach the clinics, we also have this final layer of information about therapeutic areas, indications, etc. So big numbers are great, but they're difficult to interpret, right? And this already goes into one discussion that we were having with Pascal and Brenda yesterday. So I guess you're familiar with this resource. So this is the Links 1000 from the Broad Institute. So these guys have tested something like 20,000 different drugs in 3 and 77 different cell lines, 3 different concentrations, 3 different checkpoints, etc. So this is great. But I mean, if we want to condense all this information in the form of one single vector associated to a small molecule, there's a lot of decisions that we have to do here. And one of them is trying to disentangle what's the effect that is due to the genetic background of the cell and what's the real effect that is the small molecule that is eliciting it. We have other examples, drug response, 15 different drugs, experiments done in the Broad, experiments done in the Sanger, no correlation, only for one of the drugs, right? So first conclusion is this thing doesn't work. Well, okay, when you massage the data properly, it does work, and there are very clear correlations, right? And this is an example, very clear paper that came up from Erich, Pascal, and co-workers. We have 2 interactive networks. You compare them. First things like, okay, they don't overlap, so they're both wrong. Now you have to take into account many different aspects like the space that has been assayed, the assay sensitivity, etc. And again, when you take all this into account, then it works, and we have decent overlaps. So to cut a long story short, we kind of massage all this data to create these bioactivity descriptors. And this slide summarizes 3 years of work in my laboratory, and I'm not even going to talk about it. But again, to cut a long story short, what we have now is 25 different bioactivity descriptors to describe the bioactivities of these small molecules that tell us about the chemistry itself, the targets, the networks, cell-based assays, and the clinical information. And they're complementary. Yes.
So how do they look? Are these numbers, or what are they?
These are vectors. Yes, I'm sorry. This is what you see here.
In the coordinates.
So it's 128 dimension vectors, and they're continuous vectors. So for each one of the bioactivity spaces. So this is what I'm showing here. So each row is a drug, and what you see here is one of these descriptors for each drug in one of the spaces, chemistry space, mechanism of action in space, interaction space.
They're real numbers.
They're real numbers, yes, yes. So they're vectors. This is what they are.
Somehow mapped all this into a vector of real numbers.
Yeah.
But did you say the dimension was 25 or 120?
No, it's 128. So we have 25 different bioactivity spaces, and then the vectors in each one. So the descriptors in each one of these spaces 128 dimensions.
Why 128?
Well, it's a long story. It's in public. So okay. It very much depends on the space. So if we're dealing with gene expression, so the initial vectors that we're dealing with is 18,000. Because it's for all the genes. If we are dealing with physical-chemical properties, we only have something like 18 dimensions, right? So we are kind of compensating everything. I mean, trying to keep as much variance as we can, etc. And at the end, I mean, we did lots of chess, and 128 was a good vector, basically. Yeah.
What do you do with the missing information?
What do we do with the missing information? There's a lot of missing information.
So it remains like a 0 vector?
Oh, for this, there's no missing information. This is, I'm sorry, guys. I mean, I just skipped it. I mean, this really goes into one kind of abstract representation, right? So there might be missing information in here. So this is the raw data. But then at some point, when we go from what we call signature 1 to signature 2, what we're building here is similarity networks. And then we're using node to back. So this is NLP. So there's no empty spaces in here. Yeah. So this is just to show you that some of these descriptors are fully complementary, right? That's one example. These drugs that are used in ophthalmology, whatever. So you can see that there's barely no similarity in the chemical spaces, but then there's very clear structure, both with respect to mechanism of action and interactive networks, for instance, right? This is a cool idea. Is it worth anything? So the best we can do is to compare it to things that we know, right? So in this case, it's mechanism of action and therapeutic areas, for instance. And again, you can see that in general, for all the spaces, molecules that have similar descriptors tend to have similar mechanism of action and belong to similar therapeutic areas, right? So there is some information that we're clearly retaining in here. So, as I said, so we're developing this from experimental information for 1 million molecules, right? But we don't have complete information for all these molecules. So this is what happens here. So we always have chemical information. Most of the times we have information about targets. We barely have any information about clinical signatures in here, right? So what we have done is try to infer the missing signatures for these 1 million molecules. So imagine for compound A, we always have the chemistry and we have some sparse signatures. So we can complete using Siamese networks, all the missing signatures. And then of course, we can attach a kind of reliability score to each one of these inferences. It's not really reliability, it's something that we call domain applicability, right? And now we do have a complete set of 25 bioactivity descriptors for each one of these 1 million molecules. And this is already enough to build deep networks that go directly from the chemical structure to the prediction to the inference of these bioactivity signatures, right? And this allows us to go wild and to predict these bioactivity descriptors for any molecule of interest. This is just one example. We're doing it for drugs, for human metabolites, bacterial metabolites, compounds in plant extracts, or compounds in food. Does it have any value? It does, right? So this is just one example. So this is how drugs, for which we know the mechanism of action, cluster in the space if you only consider their chemical structure. And this is how they cluster if you consider their bioactivity signatures, right? So when you see this here, so we're comparing it to the targets, mechanism of action, therapeutic indications, or diseases. So whenever you see blue in here, it means that the bioactivity signatures can produce more coherent clusters. Whenever you see red, it means that it's the chemistry that works better, right? So you can see that there is really an added value in using these bioactivity descriptors. And of course, we can use this, go wild again, and start playing with machine learning, etc. This is just a few benchmarks of different complexity, clinical toxicology, side effects, just penetration of the blood-brain barrier, etc. And then in general, you can see that whenever we use bioactivity descriptors, we get better performances than when we only use chemical information. Not always. The more complex the task, clinical toxicology, the better we are. The simpler the task, like binding beta secretase or the protease of the HIV, we get similar performances than using just chemical information. Small molecules. How about biology? This is a slide especially for you, Pascal. So I defended my PhD in year 2000, right after the first draft of the human genome came out, right? Then I started my postdoc and I was very happy because this was in the early 2000s and we had the first genomes, the first transcriptomes, proteomes, interactomes, people here in the audience, and it was a lot of data, but we could cope with it, right? We heard talks yesterday about time and space transcriptomics, right? So we have in different environmental conditions, extra levels of regulation, personalized genome. So we cannot cope with all this data anymore. My brain cannot cope with it, right? So we're talking about more than 200 petabytes of data stored at the EBI in more than 1,700 databases. So we need a machine to interpret this for us, not always to understand it, but at least to try to make some sense out of it. And since we were happy with the results of the chemical checker for small molecules, we are trying to, or we try to do something similar, but coming up with specific descriptors for biological entities, right? So the first thing that we did was to collect this gigantic knowledge base graph that contains information extracted from more than 150 different databases around 12 different biological entities: genes, cells, tissues, diseases, compounds, etc. 67 different associations. So a gene is dysregulated in a certain cell, or is mutated in a tissue, or is related to a disease. And overall, we have more than 30 million ages in here. And what we can do, I mean, we build this gigantic knowledge graph, we can explore it using random walkers, right? And then involving any type of relationship among these 12 different biological entities that we want. And then again, we go and play with NLP algorithms, right? So that we can transform this corpus of random walks into vectors, right? And at the end of the day, what we have is over 1,000 different descriptors associated to each one of these 12 biological entities that are context dependent, right? So classical one, so you have your favorite protein and you can encode it using the amino acid sequence. But now this descriptor can be different depending on whether you're interested in the sequence, you're interested in the interactions that this protein is associated with, or the transcriptional changes, or whether it's a target for a given disease, etc. So we can describe these different biological entities according to 1,000 different context-dependent ways. And what next? We have vectors chemistry, vectors biology. We can merge everything. And this is what we're doing. We're working towards what we call a general connectivity, where the idea is that we have our favorite biological signatures of interest that either come from the biotech or data that you are generating in your lab. And we have bioactivity signatures for small molecules, right? So we can start making matches and looking for small molecules that are able to match a given signature or to revert this signature. Examples. This one is related to Alzheimer's disease. So as you can imagine, most of the data that populated both the biotech and the chemical checker comes from cancer cell line panels, right? So we wanted to see whether we could extract some information outside this realm. So what we did in the lab is to use these SY5Y cells that are kind of neuron-like, and then we introduce some mutations that are related to Alzheimer's disease. So we engineered these cells in the lab, right? So the chemical checker, anything, has never seen these cells. And then basically we run some differential gene expression experiments, wild-type cells, mutated cells, and then we look for compounds in the chemical checker that were able to revert these differential gene expression signatures, right? So look for compounds that are able to move back the sick cells into healthy cells. Okay, this is just to show that these engineered cells really work as they are expected, so changing the ratio between AB40 to AB40, etc. And these are the results. So we did find, indeed, 3 compounds, uncharacterized compounds to approved drugs that are able to revert this transcriptional signature related to Alzheimer's disease. So we see here, so we are pushing up the genes that are downregulated in Alzheimer neuron-like cells, and we are downregulating the ones that are upregulated. And this is not just random genes, right? So we're hitting the genes that are really related to Alzheimer's disease. So this guy in here is involved in vesicle endocytosis. This one here is the glutamate receptor at the synaptic membranes, etc. Not for all of them, but we're able to revert something like 35 to 50% of these genes. Cell-based assays, how about in vivo? So what we did in here, we were playing with 3 different mouse models related to Alzheimer's disease, 2 knock-ins, 1 transgenic. We took the hippocampi of these mice, and then we ran full transcriptomics and proteomics experiments. We played a little bit with the standard bioinformatics. We identified modules, and then what we saw is that there were some modules, both at the transcriptional and at the proteomics level, that remained very stable as the disease was progressing, but was firing up, as in healthy aging, right? So these are mice 3, 6, 9 months old, but that are healthy, but they were firing up in the Alzheimer mice, right? So here we played exactly the same game. So can we find any drug that is able to revert this transcriptional or proteomics module? And as you can imagine, because there was way too complex for all the genes, and there was a lot of heterogeneity. So we looked, so in the first case, it was very well-controlled experiment. It was exactly the same neuron with a couple of mutations. Now here we're talking about the whole hippocampus, right? So we really had to do this previous step first. So, and then again, the other thing is that we're playing mice here, not cells, which means that we cannot just feed them with any compound that we want. So it has to be approved drugs or something, right? So this is the game that we played. And then after, so basically using the chemical checker from the initial 1 million compounds with the drugs, we ended up with 80, and that we saw that belonged to mainly 2 different groups, right? The non-steroidal anti-inflammatory drugs and antihypertensive. So we selected 6 of them that had never been related or tested for Alzheimer's disease. And to cut a long story short, we treat the sick mice for a month, and then we run some behavior experiments, right? In this case, it was a novel object recognition. So I don't know whether you're familiar with it, but you just put mice in a cage with one object. So the mice get familiar, then you replace the object or put another one. And then the healthy mice recognize that there's another object, a new one, and it's curious and spends more time exploring that one. The Alzheimer mice just don't remember and spend the same time exploring the old and the new. This is what you see in here, right? So the healthy mice spend 60% of the time exploring the new object. The sick mice spend 50% of the time on each object. So 4 out of the 6 compounds that we tested were indeed able to revert this behavior. And then when we went to the molecular signatures, we saw that indeed we are reverting the gene expression patterns. We didn't check the proteomics, but we were reverting the gene expression patterns in these mice. 3 of the 4 compounds were kind of pushing down the overexpressed genes, but there was only one, that is the one that worked best in behavior experiments as well, that were able to lift the downregulated genes in the mouse model.
Can you say about what you mean by reverting? So you want to get back to normal?
Exactly. So we want, so the genes that are upregulated in Alzheimer mice with respect to the healthy mice, we want to put them down to the same level, and the ones that are downregulated, we want to push them up. You can see that we never achieve a total effect, right? So this is the expression. So we can able to push them down a little bit, never to the real basal state, right? In some cases we are, but not in others. But in any case, this seems to be enough at least to see an effect from the phenotypic level. This is just, I don't know whether I have time. Yeah. So this is just another example, simpler one probably. So what we wanted to do here is to mimic one signature. So you know that the biodrugs are very trendy and very powerful, right? So monoclonal antibodies, etc., but they're expensive to produce, difficult to handle. So our rationale here is like, if we can find a small molecule that does the same job than a monoclonal antibody, this might have some value, right? So this is just one example. We tested several. So this is, we're trying to mimic the activity of the daclizumab. So this is an antibody that is in use to avoid rejection upon organ transplantation, right? And what it does is it binds to the interleukin-2 receptor and it avoids the signaling down, right? And it was firstly discovered using PMBC cells. So what we did in this case is like, okay, so let's kill genetically the IL-2 receptor. Then again, we run this differential gene expression experiment. So cells, healthy cells and cells with the knockdown IL-2 receptor. But in this case, we're not only interested in the transcription, we don't care about the specific target. So we want anything that will achieve the same network effect globally, right? And then we look for compounds that were able to mimic this signature of the knockdown, the genetically knocked down IL-2 receptor. To cut a long story short, we came up with 19 compounds, and 14 out of them we were really able to prevent proliferation depending on IL-2. Right? This is just one experiment and one cool example of the power of this network approach applied to the bioactivity signatures. So this is for compound 20, uncharacterized compounds. This is what's happened with the cells that do not have interleukin-2. So there is no proliferation, right? So we add interleukin-2, but no drug, and then cells start proliferating. We add the antibody, and then the cells stop proliferating. When we add this compound 20, we are able to stop proliferation in a very nice dose-dependent manner. But we know that downstream of interleukin-2 receptor is STAT5 that gets phosphorylated. And then this is what we see here, right? So we see phosphorylation when there's interleukin-2 but no drug. When we add the antibody, phosphorylation stops. When we add this compound, phosphorylation of STAT5 is unaltered. So whatever it is that we are doing, that this compound 20 is doing, is achieving the same effect, that is avoiding proliferation depending on interleukin-2, but is not affecting directly the IL-2 receptor or what is immediately downstream of it. So this is why we call it a systemic approach, right? So we're achieving the same effect, but touching a different target. And this is just to show that this is not a nuclear bomb. So we're avoiding proliferation depending on interleukin-2 only. So this is kind of my wrap-up slide. So I think, despite the boring title, Pascal, I think that I've shown you that we have created this chemical checker that is a repository that contains experimental bioactivity information for about 1 million molecules. And then we have developed these bioactivity signatures or fingerprints that we can use to extend the similarity principles to different levels of biology, right? Then using all these, we have developed what we call this signaturizer, that is now that we can infer these bioactivity signatures for any compound of interest. And we're merging it with these biological descriptors that we englobe in what we call the biotech. And all together, we can start playing this match revert thing between biological descriptors and small molecule descriptors in this kind of general connectivity. I showed you a couple of examples of Alzheimer's disease, taclizumab. We have a few more that are published and some others that are coming. And if anything, I always think that this is kind of this parable of the Indian blind man and the elephant, right? So I guess you're familiar with it, but basically it's these 5, 6 Indian blind men that get into the jungle and then they bump into an elephant. So this guy in here, they're blind, of course. So say, look, I found a rope. Say, no, what do you mean a rope? It's a wall, I cannot pass. No, it's a tree because I can hold the trunk, right? And then the idea here is that they all have partial information. So if instead of using only one type of descriptor for small molecules or for biological entities, we can combine different types of complementary descriptors, maybe we can have a global picture of what small molecules or the different genes, etc., will do. And this is the most important slide. This is my group, close to my place. And basically, most of the work that I presented was done by Miquel Duran in collaboration with Eduard Pauls and some technicians in the lab. And thank you very much, and we'll be happy to take questions. There's
really don't know the mechanism of action in a way. So how about side effect and a kind of unforeseen,
Yes, I mean, it very much depends on the type of descriptor that you use to mimic or to revert this effect, right? If you go for mechanism of action or target, in principle, you should be mimicking this effect exactly. If you go for something more global, anything could happen. Then, of course, what you can do, and we're doing, is start playing with combinations of descriptors, saying, okay, so I want a molecule that reverts or mimics this effect, but are not close to these other molecules that I know are causing trouble.
So in a way, what means to mimic as a molecule is the fine way to use it as you are.
What means to exactly, yes, is the different criteria. So in terms of our descriptors, what it means is finding molecules that have close descriptors to the things that you want. But the combination of these different bioactivity spaces, it comes by the user. In the case of Alzheimer, in the cells, we only use transcriptional data. In the mice, we use transcriptional and proteomics. With the Clizumab, we use transcriptional proteomics and networks. Sorry, not this.
So, okay, so a couple of questions. One is why, is there any reason why you use 1 versus 2 versus 3 data types, or just it happened that way?
What do you mean 1 versus 2?
So you just said now for Alzheimer's, you're going to use only transcriptional, for something else transcriptional, and
Right, it depends on your experimental data. So for the cells in Alzheimer, we engineer the cells and we only run the transcriptional experiments. So we can only use this data. So if we're talking about any disease that is well characterized and you have lots of data, you can use everything, of course.
And the second question is, in your chemical checker, you have, I don't know, if you go to the previous slide, you have 1 million.
1 million compounds, yeah.
No, next slide, like the conclusion slide. So you have 1 million compounds, and then you have their signatures and like these vectors. Do you also provide the raw data out of which you made these vectors?
Half.
Only
Half and half. So we cannot do it. So what we have, I mean, I'm sorry I didn't go too techy here, but we have different types of signatures, right? So the first signature is what we call signature 0, that this is just a very easy pre-processing of the raw data.
But
right?
you don't provide raw
We
data.
don't provide these. No, the raw data we cannot. We don't want and we cannot.
You can.
First, we don't want because we have to credit the people that is maintaining the primary databases and that did these experiments, right?
Yeah.
And the second one is that some of this data comes from databases that are protected, right? So it's like everything is public for academics, but not everything is public for industry, for instance. And then you can always provide the transform data, our signatures, but you cannot reproduce the raw data. But if you use the signature 0, this should be very, very similar, because this is just a big matrix. We're not condensing anything into vectors or anything yet. So it's just a big matrix saying all these compounds, all the features, in the case of gene expression, the 18,000 different vectors, is a yes or no, or is a continuous value. So our signature 0 that are in the chemical checker and the biotech, you can download these types. But the raw data precisely we're not providing because we don't want and because we cannot. Ready?
Well, no, but just so this thing that you did with the Alzheimer. So there are thousands of differentially expressed data sets comparing diseases versus normal. So you could do it automatically for all of them, right?
But we cannot test them experimentally, and then the value is going to be pretty limited. So we're doing that in some cases in collaborations with other people. For instance, with Antonio Garcia de Herreros in the UPF. We're playing with inhibitors and indirect inhibitors of SNL1 to avoid the cancer metastasis, for instance. But the value here is to be able to validate something experimentally, and my wet lab is very smooth, so we cannot do everything. And for the bioinformatics, as I said, we're providing everything. I mean, the code and all the signatures for free. So you guys can go play it and do it with your favorite disease.
Yeah, so it's beautiful. I really love this stuff. I'm not familiar enough with the signatures, obviously, but when you go into your biological signatures that you briefly mentioned, and look at those for, I don't know, complex autoimmune disease, right? We have very good genetics there. Can you use those on the one hand to identify new predisposing genes, but then also for the ones that you already have, say, okay, this one is probably working in T cells, this other one is probably affecting the function of epithelial cells, and kind of better use the signatures and the combination, the fusion of all this knowledge to better understand the disease mechanisms that are underlying this?
Yes and no. So we are doing that, and it's coming out in the paper, and part of it is in the bioarchive already. So what we can do is use different descriptors, right? So these 1,000 descriptors, and then we can see which ones are the ones that better match the disease signatures that we have experimentally, right? And then this will give you an idea of saying, look, if we see that the descriptors that contain protein-protein interaction information are not relevant at all, then probably we cannot explain this using protein interactions. Or the transcription is very important, then this gives you some hints, right? What we cannot do is directly relate these very abstract descriptors to the genes that are bringing the information. This we cannot do. But what we can do is predict. I was saying, okay, so we see that for this phenomena, these descriptors are the most relevant, and then we can go and predict new biological entities, for instance, new genes, etc., that have similar features. And then some of these, not experimentally, but some of these have been validated in this kind of time series, right? So we're using data that was not using here, and then we make predictions, and then in a couple of months or 2 years, there's information that comes out, and in some cases we were right, in some cases we were wrong. So you can use it to understand, to get some hints of where the signal comes from and to make predictions, but not to trace down exactly to a gene or to an interaction that is going to be key for this disease. So no mechanistic, unfortunately.
Okay, I guess we have now.
Thank you.
We reserve the questions for later. Thank you. Next speaker is Jimmy S. He has to reveal his title here.